Our second Momentum awardee is Dr. Christian Landry, Professor of Biology and Biochemistry at Laval University and the Canada Research Chair in Cellular Systems and Synthetic Biology. Dr. Landry completed his Bachelor and Master's in Biology at the Université Laval before obtaining his PhD from Harvard University and completing postdoctoral training in Systems Biology and Biochemistry at Université de Montréal. Dr. Landry started his independent position as a CIHR new investigator and a Fonds de Recherche de Québec Santé junior investigator in 2009. In 2015, he was appointed Canada Research Chair Tier 2 in Evolutionary Cell and Systems Biology, and in 2020, appointed Canada Research Chair in Cellular Systems and Synthetic Biology Tier 1. Currently, Dr. Landry is also leading a pan-Canadian training program on the evolution of fungal pathogens funded by NSERC. Dr. Landry recently received the Université Laval Excellence Awards in Teaching and Mentoring, the SMBE Margaret Dayhoff Mid-Career Award for Outstanding Contribution to Research, and the NSERC EWR Stacey Memorial Fellowship, and in 2014 was elected to the Royal Society of Canada's College of New Scholars, Artists, and Scientists. The 2024 Canada Gairdner Momentum Awardee, Dr. Christian Landry, will be presenting his presentation. The title changed here, How Evolutionary Biology Brought Me to Biomedical Research. Please.
Thank you. All right. Thank you very much for having me. It's super intimidating presenting in front of so many great scientists. Also intimidating to be presenting in front of my son and my sister, I think, who are seeing me for the first time talking about science. I want to start with telling you about my journey that brought me here, because it's a little out of the ordinary. So I started in biology, and I thought when I started biology I would study big mammals. I would become a naturalist in the Gaspésie National Park, where I come from. And as an undergrad, I studied developmental biology in a cancer lab. And as a master's student, I discovered that I was interested in biology. I studied the population genetics of salmon, a species that we like in Canada. And then as a PhD student, I was still interested in evolutionary biology, and I joined Harvard University to study the evolution of sea urchins, how sea urchins become, one species become two species. And something really major happened in my life. My PhD advisor left Harvard, he went to Stanford. I didn't go with him, so I had to join a new lab. And that's why I joined the lab where they studied molecular evolution using fruit flies and budding yeast. And I started studying evolution, but at the molecular level, and that's what I've been doing ever since. And when I came back to Canada, I really wanted to know more about proteins and the way that cells work, so I joined a lab in biochemistry where I studied protein-protein interactions. And that's what made me who I am now. And everything that I've been studying in biology is under the lens of evolutionary biology, because that's the thing that I discovered in biology that made biology a little bit like physics and mathematics, where there were principles and things that explain how the diversity evolved in nature. There were main forces that we could study, and that's what made biology interesting to me. And so biologists have been studying evolution for a long time, and the main thing that we know about evolution is that thing that Darwin put together is natural selection, and we all agree this is the major engine of evolution. But if natural selection is the engine, what is the fuel of evolution are actually mutations. That's what can feed into evolution, and evolution is the algorithm, and mutations are the data. And thanks to the development of technologies that we've heard of today, we can now study mutations systematically. So surprisingly enough, the thing that we didn't know a lot about up to recently is what mutations do and how do they change the biology that makes things improving with time. So now we can use synthetic biology to create mutations, and we can use high-throughput sequencing to read mutations, and we can now associate the impact of mutations with what they are, and that changes the way that we do biology now. So the context in which we study that is in the context of antimicrobial resistance. So one of the major progresses we've seen in human history is the increase in life expectancy in humans. So it has increased more than twofold over a few hundred years. And one of the reasons why this is happening, and that's actually the main reasons why this is happening, is that we can control microbes. Many of the things you've done since this morning is actually controlling microbes. You wash your hands, you wash your teeth. All of that is contributing to making us not die when we're young. But what happens when we do that is now that we become the agent of selection. We're selecting microbes, and we're applying selective pressures on microbes, and microbes change in response to that. And of course, the context in which this is the most important right now is in the context of antimicrobial resistance. So, there are many numbers on antimicrobial resistance. I can just tell you one. People estimate that now there may be 1 million people dying every year because antimicrobials don't work. That number is going to increase by tenfold over the next 25 years. More people need antimicrobials. We have more people with weakened immune system, and we're simply more people on Earth. So we need to do something about it. And so this is what brought me to applying evolution to biomedical research. And we study something that's a bit different from what many people study. There's a lot of problems with antimicrobial resistance in bacteria. We study fungi. So I was trained on working with the budding yeast, the model system in cell biology. And many of the fungi that infect humans are super close to yeast, so we can use the tools that we know of and contribute to that. So fungi don't infect humans very often. They usually are a problem with people with weakened immune system, but we carry them all the time. If our immune system is weakened, they can invade some organs, and they become an important problem. And in spite of their importance, and that we realize now, there's a tiny fraction of all funding dedicated to pathogens that's actually dedicated to studying fungi. So it's a good timing to be involved in that type of research. So the way we see antimicrobial resistance in fungi is we see that as an evolutionary problem. So you can imagine when there's an infection, there's a population of fungus, yeast, for instance, that decreases in size. And then we detect it, we start treating with drugs, and we hope that there won't be any mutations that happen before we can decrease the population. So it means that what's happening in a treatment is a race between mutations and the impact of treatment. And if there's a mutation that occurs, then treatment is going to fail. So we set up a research program where we want to measure like this aspect of antimicrobial resistance, which is what these mutations that can cause resistance. And we're trying to understand why can it evolve so fast. So for that, we think we need to measure how many mutations in a genome can confer resistance. Because the more mutations there are in a genome that can cause resistance, the easier it is or the faster a mutation will occur that causes resistance. And then we want to know if there is resistance to one drug, is that mutation leading to resistance to many other drugs? Because it's going to change the way we treat patients. And finally, one important question in the field is, are mutations causing harms to the pathogen? Because if a mutation decreases the fitness of the pathogen, when we stop treatment, that mutation is going to disappear. It's going to be selected against. If it doesn't cause harms, then it might stay in the population and could even be transmitted. So that's a parameter that's super important. So if I summarize these things that we want to measure, so you imagine there's a resistance mutation happens in a single cell first, and then you apply selection with a drug, the frequency of that mutation increases, and then you discover that a treatment doesn't work, you might stop it. If there's a cost to that mutation, so if that mutation decreases fitness of the fungus, it's going to disappear. If there's no cost, that mutation can stay in the patient, or it can even be transmitted. And then the other important factor is, now you can change treatment. You can choose another drug. But if that same mutation causes resistance to many drugs, there are not many options you have. In fact, you might not know, but you might not have any options if a mutation causes resistance to all of the drugs that are available. So the way we want to quantify the impact of these mutations is to do that systematically in the genes that are known to cause resistance. So what we do is that we take a gene or a protein of interest that is involved in antimicrobial resistance, and we make libraries of mutants in those genes, and we measure the impact of all these mutations on resistance to the drugs of interest. So we've been doing that for the past few years, and there are like 4 classes of drugs that are used to treat fungal infections. 3 of them, they specifically target proteins. These proteins here that we've been studying. So we've been doing these kinds of experiments I will tell you about in a few minutes. So I'm going to tell you just about one story that we just published, but the idea is the same for all these studies that we recently started doing. So what we do is that we create diversity. We take that gene of interest, we create all the possible single amino acid mutant in that protein, and then using yeast as a model system, the Baker's yeast that we know of how to work with in the lab, we can combine all of these mutants in a single flask. We grow them, we can compete them with drugs or without drugs. And then we use, that's where high-throughput sequencing becomes useful. We can follow the frequency of all these mutants in the competing populations using high-throughput sequencing. And then we know we can track all of these mutants in the tube, and we can see if we're growing that population in the presence of drugs. We can see that mutants that cause resistance are the ones that increase in frequency. They are positively selected. And the ones that decrease in frequency are the ones that actually perturb the function of the protein without conferring any advantage. So we can do that with proteins like this one. This one is the target of a class of drug that's called azoles. So that's the most frequently used drug to treat fungal infections. And it is an enzyme that processes this molecule here, and there's an active site in the middle. And what azoles do is that they block this active sites. And resistance mutations, they can prevent the drug from coming inside the protein, and that's how they confer resistance. So what we can do is that we can take a gene from a fungal pathogen, express it in our model system. In this case, we take this particular protein called ERG11, and we mutated all the sites around the active site of the protein, which is basically where the drug usually enters the protein. And then we can express the gene from that fungal pathogen in the model system, and we use tools of synthetic biology to just repress the gene that exists in that species that codes for the same protein. So the cell here is going to grow, and all growth will depend on the gene from the fungal pathogen. So if we want to answer the question of cross-resistance among molecules, we can do the experiments I just told you with a bunch of drugs. There are many azoles that are used to treat humans that are infected with different fungi. So we can do the kind of experiments I told you about with all of these drugs and measure the contributions of mutations to resistance and see if there is cross-resistance. So the kind of raw data that we get is going to be shown on this screen. It's basically a dictionary of mutations, all the possible mutations in that protein. So we have here on the x-axis all the positions that we mutated in the protein. On the y-axis, all of the possible amino acids that were inserted at those positions. And then the color represents the impact of those mutations. So this experiment was done in the presence of azole. When it's green, it means that these particular mutations, they confer resistance to the drug. And brownish red means that these mutations are harmful for the protein. They decrease the activity of the enzyme, but they don't confer resistance. So we can create these kinds of maps for the set of positions that we studied in this particular protein. And then you can see there are hotspots in the protein that confer resistance. Those are presumably mutations that prevent the drug from entering, but that maintain the function of the protein because the protein needs to be functional for the fungus to be resistant. So this is the kind of experiments we do. Now we can ask, so this is a map for one drug. If we do that for all the other drugs, what kind of images do we get? We actually, surprisingly, although the molecules look different, we surprisingly find exactly the same kind of image. So we did the experiment with many drugs, and with that, we can show that there's a lot of cross-resistance. What that means is that if there's a mutation in a patient that prevents a drug from working, 70% of the time, all of these other drugs will fail treatment. So this is something super important to be measured. And also, it shows how important it would be to have this data when you switch treatment from one drug to another. Now, the second thing that I mentioned is super important to measure is the cost of these mutations. So we can measure how these mutations impact the normal function of the protein, which I am representing on the y-axis here, which is a measure of the impact of a mutation on the function. Negative means negative impact. And we can measure the impact of the same mutation on resistance. And what you can see from this plot is the following. So you see every data point, every point is the impact of one mutation. You can see all these mutations, they increase susceptibility to the drug, so they are interesting mutations. And only a tiny fraction of the mutations, they lead to resistance and are deleterious for the protein. And in fact, most of resistance mutations, 90% and more, they increase resistance, but they cause no problem to the protein, which means that these mutations, if you stop treatment, they won't disappear. They can stick around because they don't impact the fungus that much, or not at all. So this is a kind of tools that where we combine the kinds of experiments where we combine synthetic biology and high-throughput sequencing to produce data that's useful from a point of view of evolution of resistance, but also I will tell you how we can use that data practically in terms of medical application. So for the last few minutes, I want to tell you a few things that I also did in the past few years. When I got into that field, we realized that there was very little training among people who study fungal pathogens in terms of evolution. Although I told you this is a problem, an evolutionary problem, if we don't understand evolution, I think we cannot deal with that problem perfectly well. So what we did was to create this training program across Canada that started 3 years ago with many different universities. So I led the creation of this program through NSERC Create training grant. And we're about 10 PIs across Canada who work on fungal pathogens of humans, but also of plants, because there are many things that are shared between plants and human pathogens. And for the past 3 years, we've been training PhD students and postdoctoral fellows across Canada. They work together, they take classes together, we created new classes, and we train them on the evolution of fungal pathogens, also on technologies like CRISPR-Cas9, genome editing, high-throughput sequencing, and bioinformatics, so they can implement now the kind of experiments that I told you about. So far, we've trained nearly 60 fellows, and we only have 2 in the program, so I think we're contributing to the next generation of people who can help solve the problem of fungal infections. So one of the concrete and cool thing that we did with this group of people in the past few years was that we realized when we were looking for mutations that all the data that people had collected over the years on resistance mutations, they were scattered in different papers, like papers reporting a single mutations. I showed you the experiments where we can track like thousands of mutations at the same time, but we thought that mutations coming from clinical isolates were super important to report and be put together. So we took I invited many of the fellows in the program, and I said, let's spend a few weeks together. We're going to read all of these papers and collate all of the data so we can create a database that now people can use in the field, and they can go to the database instead of trying to find mutations in the literature. So we curated many papers, several hundred papers. We ended up using data from 400, a bit more than 400 papers. And now we have like thousands of associations between mutations and resistance to antifungals. So what can we do with that? So when I received the instructions for this talk, they asked me, what's the impact of your work? Most of what we do is super fundamental. That's why I chose to talk about this particular project. But here there's something super useful that we can do, and we're actually doing with public health labs in Canada. And how you can use this information is to try to assess more rapidly the susceptibility of fungal infections to antifungal treatment. So the way that clinicians do in Canada, at least, when they have a patient who's infected with a fungus, is that they take a sample, they grow the sample, they hope it grows, and then they isolate a few colonies of that fungus growing. They send it to public health labs, and in the public health labs, they grow them and they do the phenotypic testing. They say, this drug works, this one doesn't work. They call back the doctor and they tell them, you should be using this drug rather than this drug. Sometimes they just start with the standard procedures and they might adapt depending on the results. This works perfectly well, but it's sometimes too slow for some patients who are critically ill. What we're trying to do with the public health labs is to try to skip all of that growing step, and we try to work with them so we can take DNA samples directly from the patients, sequence the genome of the fungi, and then use AI-based technologies with the data that we collected so we can predict what the fungus is going to be resistant to. And with that, we hope that we can save some time. So that should take maybe 2 or 3 days, whereas this can take 2 or 3 weeks sometimes. So this is all the data that we collect can be useful in terms of medical application. Okay, just to summarize what I told you about, we are a super strong selective pressure on microbes, and microbes evolve in response. It's an interesting problem for an evolutionary biologist, but it's going to be a concrete problem for us in the future. And this is particularly true for antimicrobial resistance. And we think that by combining what we know about evolution, we can solve some of these problems more easily. And now we can make use of the high throughput technologies, some of them that were invented by people in the room, which is quite amazing to be presenting that to you. And we can study these mutations before they even occur. So many of these mutations, they never occurred in the past or were never seen. So it's like accelerating evolution in many, many dimensions at the same time. So we can foresee what can happen, and then we can act better because of that. And what is going to come from that, I hope, is that one of the major problems when we study human fungal pathogens is that it might sound surprising for some of you, but we're super close to fungi in terms of evolution. Fungal cells and human cells, compared to all the diversity that exists, are basically the same cells. So it's super difficult to find proteins that we can attack with drugs that would kill fungi, but that would not kill humans. So we already have a set of good candidates for drugs, but the problem is that they are resistance mutations. So maybe what we can do is to modify the drugs to overcome resistance mutations rather than trying to find new targets and new molecules that are completely different. Okay, so that's it. That's what I wanted to tell you. I want to thank the people who do the work. Everything we do, all the stuff that I showed you is done and led by graduate students. And the funds, importantly, the funds from granting agencies. And then there are 2 important people on this picture who are also in the room, Isabel and Alex here. They are research professionals who've been working with me almost since the beginning of my lab, for Alex and Isabel since the beginning. And we work on all projects together. And I've been able to have them in my group because of sustained funding from CIHR, for instance. So that shows that funding is super important. We can keep these people around us, and they help make great progress in science. Thank you very much.